We're going to continue with our lecture on gram-positive bacilli, and this is the second of 5 parts to this lecture. In this section, we're going to move on to some of the other gram-positive bacilli or gram-positive rods, since in the first part of this lecture we focused on the Arcanobacterium genus. Another gram-positive rod is the Arcanobacterium genus. There are 5 species that have been identified in this genus, and 3 of the species are medically important to humans. These species are Arcanobacterium haemolyticum, Pyogenes, and Bernardii. All of these organisms are catalase positive. Arcanobacterium haemolyticum has been recovered from 10 to 20-year-old patients with pharyngitis. It's been associated with causing a rash and causing the skin to slough off from hands and feet of infected individuals. It produces very small colonies and a very narrow zone of beta hemolysis around these colonies on sheep blood agar. It is lipase and lecithinase positive, and it's resistant to the antibiotic penicillin. Rothia is another genus, and the species of Rothia that has been associated with infection is Rothia equi. It's found in the soil. It causes respiratory tract infections in animals. Human infections are incredibly rare, but it has been associated with infection in immunocompromised patients, such as AIDS patients. This organism does not ferment carbohydrates, and it shows variable reactions to some of the biochemical tests. So it's not that easy to identify, and luckily it's not commonly associated with infection. Microscopically, the Rothia organism looks a little bit to be branching. It can be a partially acid-fast organism, meaning that it can be stained using the partial acid-fast staining method. Macroscopically, it looks like a Klebsiella organism. It forms a salmon pink colored pigment when it's incubated for prolonged periods of time. So over 24-hour incubation, it will start generating a pigment. Now we're going to move on to other gram-positive bacilli. We started out with the Corynebacterium diphtheriae, causing the disease diphtheria. Now we're going to move on to another somewhat common infection organism that causes infection, and that is the Listeria genus, and the species that has been associated with human infection is Monocytogenes. Listeria monocytogenes is, of course, a gram-positive bacilli, which is why we're discussing it in this specific lecture. It can be pleomorphic, meaning has various shapes, and it's considered a coccobacilli. And if you remember from our first lecture, the coccobacilli are bacilli. They're not cocci, they are rods. They are just teeny tiny rods, so they have somewhat of a coccoid or circular shape to them, but they are in fact actual bacilli. Listeria monocytogenes is beta hemolytic on 5% sheep blood agar plates. It's catalase positive. It is a motile organism, so the fact that it's catalase positive and it's motile are very important diagnostic features to this organism. Listeria has what's called tumbling motility, so the organism falls over and over on itself. So if you looked at it under a microscope on a wet mount, it would look like the organism is tumbling over and over and over, and that's how it moves from one area to another. One of the key characteristics about Listeria is it's more motile at room temperature than it is at 35 to 37 degrees. So we commonly incubate our clinical cultures in a 35 to 37 degree incubator, whether it's an ambient air incubator or a CO2 incubator or under anaerobic conditions, but we commonly use the 35 to 37 degree temperature range because that's where most of our clinically significant organisms like to grow. Now with Listeria, if you inoculate motility media and you inoculate 2 different motility deeps and you put one at 37 degrees and one at room temperature, you will see that Listeria will be much more motile at the room temperature incubated motility slant or deep than in the 37 degree incubated. Another characteristics about Listeria, and this is key, this absolutely has to be embedded in your memory. Listeria monocytogenes demonstrates umbrella motility in agar motility media. That is a key diagnostic characteristic for Listeria. It is the only organism, clinically significant organism, that I know of that demonstrates umbrella motility. This organism is a facultative anaerobe, so it will grow perfectly fine in ambient air. It'll grow in the CO2 incubator. It's not all that fussy. This is a gram stain of Listeria. You could see it's a very small rod. It is pleomorphic. There are some rods that look a little larger than other rods. This is the morphology on 5% sheep blood agar plate. Here on the left is Listeria. You can see intracellular inside some white blood cells. Now, Listeria does generate disease in humans due to its virulence factors. It produces a hemolysin, specifically Listeria lysine O. This hemolysin damages the phagosome membranes so that macrophages can't kill Listeria. So Listeria is one of these organisms that will get gobbled up by a macrophage, and it's perfectly fine. It actually will start dividing inside the macrophage and moving around and escaping from one macrophage and going in and infecting another macrophage. It does produce catalase. It is a catalase positive organism. It produces superoxide dismutase, another enzyme that's important when an organism is phagocytosed, because the phagosome can generate oxidative products which are toxic to most bacteria. It produces phospholipase C. It produces a P60 surface protein that will actually induce phagocytosis, because Listeria is perfectly fine inside a phagocyte, like I said, and will actually replicate in the cytoplasm of a macrophage. So Listeria, like I said, will get into a phagocytic cell and it will divide there, and it forms this actin tail, and it actually will start to move around really rapidly, kind of like a little rocket ship inside that phagocytic cell. It will then shoot out of that cell and then go in and infect a neighboring cell. The clinical manifestations of Listeria include a neonatal infection. Listeria in the neonate is a highly fatal infection. There are 2 different types of neonatal infection caused by Listeria. There is early onset and late onset. In early onset, this occurs either in utero, so vertical transmission from the mother to the fetus in utero, or during birth. So this results when a pregnant mother is infected with Listeria monocytogenes. It can result from an intrauterine infection. It causes illness in the baby very shortly after birth. It usually causes a sepsis. So the baby starts getting very sick very rapidly after birth. And it can be associated with the baby aspirating up amniotic fluid and then getting a septic infection that way. There's also a late onset neonatal infection. This is when the little baby gets infected, doesn't show symptoms right after birth, but several days to weeks after birth, the baby will start to show signs of meningitis. Pregnant women have to be very careful not to get infected with Listeria monocytogenes in their third trimester. So pregnant women are told never to eat imported soft cheeses. They're told to avoid cold cuts, things like that. The reason is because Listeria can actually grow in a wide temperature range. So Listeria, if it's on a cold cut, just because you put it in the refrigerator, it's perfectly fine. It will survive. And you don't cook cold cuts, you make a sandwich. So that's why, if anybody was ever wondering why, if you've ever been pregnant or you know someone that was pregnant and they told you that they couldn't eat cold cuts or they couldn't eat any soft cheeses, this is why. It's because you don't want a pregnant woman getting infected with Listeria monocytogenes. So in an adult that gets infected with Listeria, it can generate kind of a flu-like illness, maybe a little bit achy, muscle aches, headaches, maybe a mild fever. What happens is that organism will then get into the bloodstream, and if this is a pregnant woman, it can get into the uterus and infect the fetus. So Listeria monocytogenes can commonly cause premature labor, and it can lead to a septic abortion. So the mother can abort the fetus because of this massive infection. Listeria is also more commonly the cause of infection in immunocompromised individuals. So these immunocompromised individuals can get an invasive Listeria infection. This is more common in individuals like cancer patients that are undergoing chemotherapy that have absolutely no white blood cells at all. Also, very young children and the elderly are at higher risk of getting a Listeria infection. The most common manifestations are a central nervous system infection or endocarditis. The fatality rate is much higher in newborns that are under 4 weeks of age, in the severely immunocompromised, and in adults that are over 50 years of age. Naturally, Listeria is a zoonotic organism. It can be carried in mammals, birds, fish, insects. It lives in the soil, water, vegetation, so it's considered an environmental organism. It's quite ubiquitous. Ingestion is the most common mode of transmission. So again, unpasteurized milk, imported soft cheeses that may be made from milk products that are not pasteurized. There are some associated infections with meat, with cabbage, because you may not cook cabbage, cold cuts, hot dogs, those meats that commonly you might eat without cooking or cooking thoroughly. It can be transmitted directly through environmental contact, and of course vertical transmission from the mother to the infant in utero. It is incredibly difficult to control Listeria infection. This organism lives everywhere. Like I said, it's ubiquitous. It can grow in a very wide temperature range. So it can grow perfectly fine in the refrigerator at 4 degrees. It grows at room temperature, and it also grows at body temperature at 37 degrees. So again, all pregnant women are told to avoid soft cheeses, cold cuts, and hot dogs. And also, if you are going to ingest any dairy products, you have to be very careful if you're pregnant that they are pasteurized. Morphologically, we already showed it's a gram-positive small rod or coccobacillus. It sometimes looks like a diphtheroid, those Corynebacterium organisms. It can also look like a streptococcus. So a lot of times it's very difficult to diagnose an organism just based on a gram stain. It would never be done. You can narrow down what you think the organism might be using a gram stain, but then you always have to do biochemical confirmatory tests. This organism, like your diphtheroids, you can see single bacilli, you can see small little chains. They can palisade or pile up in parallel rows. You can do agar media cultures. And the thing about Listeria is it does like those cold temperatures. So if you have a specimen from an individual that might be contaminated with a lot of different organisms or some normal flora, you can actually do cold enrichment. You can take that clinical specimen, you can put it in a refrigerator for a specific period of time, and then you can culture it. By putting it in the refrigerator, it'll inhibit the growth of some of those normal flora organisms and contaminants, and it will allow the Listeria to grow, so you have a better chance of isolating your Listeria from a mixed specimen. The organism will grow on almost any laboratory media. It can be grown at room temperature or at 37 degrees, and like we said, it has enhanced motility at room temperature. It is a very small colony on blood agar. It has a slight zone of beta hemolysis, very slight, it's difficult to see. And you have to be careful to differentiate your Listeria from your streptococcus and your diphtheroids. That motility pattern of Listeria is key. The fact that it is more motile at room temperature than at body temperature, that umbrella motility, and the fact that it has a tumbling motility if you were to do a wet mount are very key features of Listeria. In order to definitively diagnose Listeria, you have to do more extensive testing, such as biochemical testing, serological based testing. It is catalase positive. It's also positive for hippurate hydrolysis and bile esculin hydrolysis, as well as Voges proscour and methyl red. It also is positive for the CAMP test. So if you remember, when we were talking about streptococcus, we talked about the CAMP test, where you streak Staphylococcus aureus down the center of a blood agar plate, and then you take your streptococcus organism and you plate it, streak one line perpendicular to the Staph aureus. And if you have your Streptococcus agalactiae, you will have an enhanced hemolysis right at that juncture, and it'll form an arrow. Well, with Listeria, it's the same thing, but instead of generating enhanced hemolysis in an arrow shape, it forms enhanced hemolysis with a block shape. So there's a square of enhanced hemolysis at that perpendicular juncture between Listeria and your Staph aureus. So remember, there's your CAMP test. Traditionally, you have an arrow, but Listeria doesn't always make a nice arrow. It tends to just make a little square. This is your umbrella motility. So on the left is classic umbrella motility of Listeria. So the tube on the left is positive, the tube on the right is a negative control. So if you can see, there is a haziness in the inoculum line in this motility deep agar. And then on the top, it looks like a parasol where the organism is making an umbrella over that inoculation line. Here it is again, and again, this is classic look of Listeria at room temperature. So if we were doing a laboratory right now, we would actually do this test where all of you would take your motility deeps and you would inoculate them. You take a needle, you take a colony of what you think is your Listeria, you put your needle straight in, as straight as you can go, about 3/4 into the deep, and pull straight up because you don't want to move it around a lot. Motility media is an agar, but it tends to be very soft, so you don't want to move around that inoculum line too much. You would incubate one at 37 degrees and one at room temperature. And the one at room temperature will give you this beautiful parasol or umbrella looking motility at the top of it. Here's Listeria inside some white blood cells. This is a clinical specimen. So whenever I say a clinical specimen, I mean a specimen that came directly from a patient. So that would be like a spinal fluid. You might get a spinal fluid in and have to do a gram stain of that spinal fluid. And if someone had an infection, you'd actually see the white blood cells and you would see your organism commonly associated with those white blood cells. We're going to move on to part 3 of our gram-positive bacilli lecture.